This November, we're celebrating the scholars that you've probably never heard of, but definitely should have. Professor Jennifer Doudna didn't set out to change the world. She was studying how bacteria defended against viruses. This was relatively basic research, the kind struggling for funding because people ask, what's the point? Then she and Emmanuel Carpentier figured out CRISPR, a tool giving humanity the power to edit DNA with unprecedented precision. Jennifer Doudna was born in 1964 in Washington, D.C. She grew up in Hawaii. She became fascinated by science after reading Watson's Double Helix as a child. She earned her PhD from Harvard Medical School. Then she built the molecular messengers carrying genetic instructions. Then in the late 2000s, Doudna began collaborating with Emmanuel Carpentier. They were investigating a strange immune response that some bacteria use against viruses. When the virus attacks, the bacterium captures a piece of the virus's DNA and stores it in its genome in a region called CRISPR. Same virus attacks again. The bacterium uses the stored sequence to recognize and destroy the invader. The bacterium does this using molecular scissors, proteins that cut DNA at very specific locations. Doudna and Carpentier realized if we could program these scissors to cut anywhere, we'd have a tool for editing genomes with extraordinary precision. And then in 2012, they published their findings. CRISPR-Cas9 could edit genes in any organism. This was a revolutionary application. Applications came quickly. Disease-resistant crops, new treatments for genetic diseases, studying gene functions by turning genes off. But CRISPR raised enormous ethical questions. If we can edit human genes, should we? The technology works on embryos. You could theoretically edit future children's genes, remove genetic diseases before birth, but also you could edit it for non-medical traits. Eye color, height, intelligence. Where will we put this line? In 2018, Chinese scientist Jiang Kui announced he'd use CRISPR to edit twin girls' genes, making them HIV resistant. He crossed ethical lines without proper oversight, any informed consent, or any thorough safety testing. He was sentenced to just 3 years in prison, but the damage was done. Technology had been used recklessly. Doudna had grappled with these questions for years. She didn't develop CRISPR to edit human embryos. She was studying bacterial immune systems. But now the Pandora's box was open, and she feels the responsibility to shape how it's used. She's called for international guidelines, public discussion, and caution. Scientists can't just create powerful technologies and just simply walk away. They must engage with ethical implications. In 2020, Doudna and Carpentier won the Nobel Prize in Chemistry, one of the fastest journeys from discovery to the Nobel Prize, just 8 years. The committee recognized CRISPR was already transforming biology and medicine. Doudna got recognition, they won the Nobel, so keep learning and keep questioning.